From millions of wet specimens underneath Chicago's Field Museum,
It's kind of like a library, but jars of fish.
to man-made lava that mimics dangerous flows in nature, America's museums and labs don't just display history, they create it. Together, these institutions contribute more than $50 billion to the U.S. economy every year and drive billions more in scientific research. But every time the federal government shuts down, facilities like these grind to a halt. This video shows you how scientists acquire, preserve, and unlock the secrets of life on Earth. The Field Museum acquires its specimens in two ways, either through donations, or sometimes researchers go out in the field to strategically euthanize specimens, like this common water snake.
If you manage to grab one, they do not hesitate to bite.
Sarah's research relies on new and old specimens to see how changes in habitat have affected the species over time. And the first thing she does with a new one is grab its DNA. This is a fairly new step in the process, since DNA wasn't really used the way it is now until the 90s.
It's not impossible, but it is much easier to take tissue samples from fresh animals than it is to take them from preserved animals and get really good results.
She uses scissors and forceps to collect the DNA sample, first sanitizing them by burning away any random DNA so the results aren't mixed.
It does get so hot though, then I have to dip it into some ethanol so that it doesn't essentially sear the animal as I work on it.
Sarah takes the sample from the inside of the snake so she doesn't mess up what it looks like on the outside.
The way I'm cutting is so that if somebody comes along, they're still going to be able to count these scales.
Plus, that's right around where the liver is located.
And there's the liver, right there.
It's Sarah's favorite tissue to collect for DNA extraction because it dissolves easily.
It turns everything pink. It says SR1291. I double check my tag is SR1291.
Then the DNA goes into these massive liquid nitrogen freezers with thousands of other DNA samples. Now she's ready for formalin, the liquid that preserves the tissue and keeps a specimen frozen in time.
It's sort of like embalming it, just like a person at a funeral home.
Sarah has to keep in mind what info she needs now and what researchers might need in the future, like the sex of the snake.
It's always good to see if it's a male snake, if you can find the hemipenes and pop them out.
Sarah will pose the snake so you can see the visible penises from outside the jar without even opening it. In this pose is how it will stay for the rest of its afterlife. The coil doesn't just look snaky.
There is some art to it. You can stack lots and lots of them on top of each other in a jar. 1, 2, 3, 4. I've got 5 snakes here, and there's another probably 6 in this jar.
Last step in this part of the process is tucking it in under a formalin-soaked paper towel. It keeps the snake saturated without having to fill the tub.
Good night, snakey.
Over a few days, the formalin will set into the tissue, leaving the snake fixed in place.
Almost like you're holding a rubber snake.
Larger animals might need more than a few injections, like this catfish Caleb is working on. Calculating the amount of formalin needed is mostly based on experience and feel. Too little and your specimen will start to decay and get floppy. Too much and your specimen will bloat and become disfigured.
You don't want to make the belly do this because you've pumped it with so much formalin.
Once Caleb is confident his catfish is sufficiently full, he'll move it into a tank of even more formalin to soak for about a week.
We're going to add a bit of cheesecloth just to make sure that no parts of it are sitting outside of the formalin.
After the formalin, the team switches over to alcohol baths for long-term preservation, like with this Komodo dragon. The alcohol is less toxic than formalin, so it's safer for researchers in the long run. And the specimen doesn't change much while it sits in its final resting tank, just the color of the liquid.
Especially large specimens, they'll release a lot of debris and fatty oils that were stored in their body, and it will leach out into the ethanol, and that causes a lot of the discoloration. It's still doing its job in keeping the animal preserved.
Most specimens in the wet collection are kept looking as lifelike as possible, but others...
We can clear away all of the tissue, stain the bones and stain the cartilage, and we can end up with just a skeleton that we can put under a microscope.
Extra small fish have extra small bones that are difficult to keep track of. So instead of isolating the skeleton, this method keeps it contained but visible inside the body. First step is dyeing the specimen blue. This specific blue dye is attracted to cartilage, and the red dye clings to calcium. A few days for each is typically enough to lock in the dye. The next step is to clear the fish.
We use an enzyme called trypsin that digests proteins and break them down, but it leaves the collagen that holds everything together,
making the fish completely see-through. Finally, he dyes the bones red.
One of the advantages of clearing the fish and then putting it into the red dye is you can keep an eye on it to see how dark it's getting.
The whole process can run a few days to around a month. Done right, and your final product are these almost alien-looking specimens.
It's kind of like Jell-O. And you store it in glycerin in the end because glycerin and collagen have the same refractive index, or the way that light passes through.
These specimens go right into collections alongside all the opaque ones. So researchers can access them when all they want to see is bones and cartilage.
You can put this under a microscope. You can move bones around and see how one bone moving affects other bones nearby.
Entirely new species have been discovered this way, like these two fish species that are identical on the outside.
But when you clear and stain them and you look at their bones, you can actually see that there are differences in their skeletons between species.
New species can hide on the shelves for decades.
So here we go.
Like this spider-tailed horned viper, kept under lock and key. It was originally collected in the 1960s, and researchers first thought it was a different species of viper with an abnormality.
This weird parasite or a tumor.
But then,
in the early 2000s, some herpetologists came along and they said, I think it's a whole new species altogether.
Once a second one was found, they compared it to this one.
And the species Pseudocerastes urarachnoides was described from this very individual, this holotype.
But specimens like this one are only helpful if you can find them.
It's kind of like a library, but jars of fish. Different families of fishes have numbers, and then within each family, they're arranged alphabetically by genus and then by species.
There's even a field in the database for noting when and where a specimen was last seen.
So if someone asks for it, we know that, well, at least at this date, it was spotted on the shelf in the collection.
And once you find it, it's not always as simple as pulling a jar off a shelf.
There are 883 frogs in here.
And each one has its own ID number. So if a researcher wants to look at a specific frog,
I have to sit here and just pick each one up one by one, be like, no, not this one. No, not this one. Until you find the right one.
These frogs used to be stored separately, but at times,
We've definitely run out of space. They combined jars of the same species, but collected by different people, collected from different places, collected at different times. But then you end up with the same problem I've mentioned here.
Getting rid of stuff is not an option. Old specimens can be especially valuable.
This is the oldest specimen we have in the amphibian and reptile collection.
Sarah has even developed a technique for recovering DNA stuck inside them. The process is similar to getting DNA from fresh tissue with a little extra work.
That extra work includes heating it up real high and trying to digest it and pull the DNA away from the formalin over a much longer period of time.
But it can be hit or miss.
It could be anywhere between 0% success all the way up to like maybe 60 or 70% success. If all you have is a 100-year-old snake in a jar, you might as well give it a shot.
Like library books, some of these jars can sit untouched on their shelves for years. But all it takes is a curious person to crack one open, and our understanding of the natural world can completely change.
I always think whether it's DNA, whether it's taking the whole specimen to preserve, this thing then didn't just die in vain. It lives on in science forever.
You're looking at a giant sloth pelvis being chipped out of the Brea Tar Pits. It's been hiding here in Los Angeles, perfectly preserved for anywhere from 25 to 45,000 years. Scientists have spent more than a century excavating some 4 million specimens from the pits. But why keep adding to an already huge collection? Because each new fossil helps fill in the gaps on a 50,000-year timeline of life in the region, spanning to the end of the Stone Age. We went to the Brea Tar Pits to see how specimens are discovered, cleaned, and pieced together to build that record. The process kicks off with a sticky scavenger hunt. There are around 130 pits at the tar pits, which is technically a misnomer.
We don't technically have tar, we don't naturally have pits. What we actually have is asphalt, which is the crudest form of naturally occurring oil.
Some tar pits are still connected to their oil source, like this one.
So right now I'm in pit 91, which was the 91st pit. I know, fancy naming schemes.
Others become disconnected from their oil source and dry out. Huge dry chunks of the deposit are easier to move into separate boxes for excavation.
Right now I'm in box 13, which is 13 of 23, as you might be able to guess. This particular box has easily tens of thousands of fossils in it.
The excavation process isn't as simple as pulling a bone out of some goo.
So sometimes I'll find a fossil and be able to excavate it out in like 15 minutes. And sometimes I'm looking at a fossil for several months, definitely not dreaming about it, teasing me.
Take this saber-toothed cat skull, which has been weeks in the works. Based on 16 years of experience, Laura can estimate how many fossils are in a given deposit. To start her search, Laura first sets up a makeshift grid. This will help Laura keep track of where and in what position fossils are found, since the asphalt is too dense for traditional radar to provide initial images.
It's eyeballs and elbow grease.
Then she begins the meticulous process of carving the skull out of the matrix that encases it.
If I am confident that I'm not going to find fossils, I'll have things like hammers and chisels that I'll be able to get in there and move some of those things away.
This is where it gets tricky. Asphalt actually protects the bone's organic matter. This also makes them more fragile than traditional stone fossils.
Around the fossils themselves, we'll start with very small tools, something like this dental pick, which yes, is sourced from local dentists. Thank you for your donations. And sometimes I'm using it the same way that a dentist would, with the tip of it. And sometimes I'm using it more like a clay sculpting tool, kind of pulling sediment away from fossils. And then sometimes you'll also see me using a natural bristle paintbrush. I don't want to use plastic bristles because the oil will actually eventually degrade those bristles and they'll kind of melt a little bit, which isn't good for anybody.
Excavators tailor their strategy to preserve not only the delicate bones, but also the matrix itself. It's filled with valuable microfossils.
So we're going to pour that chemical solvent in there. So this particular one is a Novec 73DE, and so that's a chemical degreaser.
The solvent dissolves the asphalt and leaves behind treasures.
This one is the end of a tibiotarsus, or a drumstick. This one's bigger than a chicken. And we also have plant fossils as well.
Once the matrix is collected and surrounding sediment is cleared,
so I should be able to just kind of gently release the fossil from this tangle and send it on to the laboratory,
where preparators will put in even more hours into this one saber-toothed cat skull. To make sure our skull is shelf-stable, preparators start with a more meticulous asphalt removal.
I would say that this could probably take me up to 10 hours in total to complete.
Stephanie uses more degreaser and a collection of cotton swabs, foam applicators, brushes, and picks to surface clean the skull.
When we have areas where there is a jagged bone exposed, we prefer to use the foam tip applicators because there's no grabbing of fibers that could damage the fossil or transfer too much fiber onto the fossil. It's very much like painting almost.
The asphalt permanently stains the fossil a deep brown.
You will notice that as I work through this, the tool itself is changing color and becoming darker.
Now, her goal isn't to deep clean the specimen. It's to prevent deterioration over time. So some debris stays behind.
We leave the internal matrix in the skull because it provides structural support and stability.
If a fossil is damaged or comes out of the pit in pieces, it can be fixed.
So this is a canine from box 9, and we prepared this bit in September last year and sent it through to collections. And then in May this year, we found the tip, and when we put it in there, it was like a puzzle piece. So this is the lower jaw or dentary of a young direwolf that we have over here.
She uses a little adhesive and sometimes Japanese kozo paper to secure and stabilize the broken bits.
So kozo paper is a Japanese archival paper. You'll notice that I tore it, and we want to have these fibers. Think of when you walk into a spider web. It just feels like it's everywhere. They migrate and then catch onto the specimen and really anchor well onto it.
And the final step is covering the entire specimen in the same adhesive used for repairs. This seals and protects the organic material. If somewhere down the line a researcher needs to access that material,
Because we use acetone, it makes the whole process reversible.
after preparation,
The specimen there gets its own individual catalog number and then forms part of our already amazing collection that spans over 3.5 million specimens.
We have somewhere in the nature of at least 2,500 saber-toothed cats. So for instance, all of these drawers here are full of nothing but left upper arm bones of saber-toothed cats from just one of our about 130 deposits.
beyond saber-toothed cats, the La Brea collection contains around 4 million specimens from the last 50,000 years. And the biggest extinction event since the dinosaurs, the Ice Age extinctions, ramped up 12,000 years ago.
The majority of large mammals on Earth disappeared during this time. It's been really hard to figure out exactly when because we just don't have enough fossils.
These bountiful tar pits are an exception.
This is the only place on Earth where we have a time transgressive record over a 40,000 year period of many of these species. And this allows us to do research that you can't really do at any other fossil site.
Along with the when, researchers can answer questions about why species went extinct too.
Was it lack of water sources? Was it an area getting too hot or too cold? Was it increase in fire? Was it something to do with the arrival of humans on the landscape?
Some unique specimens can tell us about an entire species' behavior, almost entirely on their own. Take this elderly saber-toothed cat with hip dysplasia. The fact that it exists at all
is an indication that this species was probably social, because individuals with injuries or pathologies like this probably wouldn't have been able to hunt for themselves and probably relied on a social group or a family group to support them.
While the lab pieces together specimens, collections pieces together data.
Understanding what sort of processes might have been happening during the last major extinction event could be really important for helping us to weather some of these unprecedented in kind of human history impacts that we're experiencing. And there is basically no more important question to be asking today.
Knowing how creatures lived amidst climate change in the past might help humans learn to navigate extreme conditions in the future, like wildfires or droughts. In this case, the more we have, the more we know. And there are no plans to stop digging anytime soon.
The Smithsonian National Museum of Natural History holds over 148 million specimens, and nearly all of them are hidden away in secure collections.
Something that people really like to see are shark jaws and all the shark teeth.
We should be able to open any of these drawers and blow your mind.
Nearly everything in this archive is available for scientific research to solve real-world problems, like helping planes avoid birds and protecting the country from invasive murder hornets.
Their head is very robust, bright orange. That's pretty distinctive.
Now, the Smithsonian is racing to build a digital Noah's Ark.
I do think that a lot of the species that we have represented here are going to be extinct, and collections like this are going to be the only place where you can find any evidence of their existence.
It's a DNA library of all complex life on Earth, supercharged with high-resolution cameras, AI, and robots.
And so what used to take us a month, we can now do in less than a day.
This data could unlock new medicines, fuels, and ways to grow food. So how is the Smithsonian future-proofing its collections? And what mysteries hide behind locked drawers?
We can't talk about this. Oh, I could tell you, but then I would have to kill you, so.
You can walk through all of the exhibits in the National Museum of Natural History in about 2 hours. But it would take days to tour the hidden collections that make up the rest of the space. More than 1.4 million square feet spread across the museum and the off-site support center. All the facilities combined are about the same size as the U.S. Capitol building.
Museum collections are incredibly important for understanding what was there, what has changed, and what might be valuable to restore.
The number of objects in the museum has grown exponentially since it was established in Washington, D.C. in 1910. New specimens arrive here in 3 ways. First, through donations, like this 8,000-pound crystal from Arkansas. Second, expeditions. That's when scientists go into the field and bring samples back to Washington, D.C. You may recognize the name of the explorer who bagged this one.
This label of the Harpy Eagle was collected in 1902, and if you turn it over, it says, presented to Biological Survey by President Roosevelt. Can you see that? That'd be Teddy.
Third, the museum can purchase specimens. However, this is rare. That's because the museum doesn't have a budget for it. The funds have to come from a private backer. In recent years, that's made it hard to compete with wealthy collectors for things like fossils.
After the great success of the Jurassic Park movies, every billionaire and tech bro in the country wanted to have their own private dinosaur.
The process to preserve and store a new specimen varies depending on what it is. Some animals get put on ice. Others stay fresh in alcohol. The museum's wet collection is filled with tiny jars and enormous vats, holding creatures like this stonefish.
Most venomous in the world. Whoops.
Fossils have to be painstakingly removed from layers of ancient rock, called matrix.
This is a carbide needle that a very dear friend of mine carved, so this is sort of like a special needle that nobody else is allowed to touch.
Specialists remove the muscles, oil glands, and other soft tissues from the birds.
We want to remove as much of the muscle that we can. We're trying to make a specimen that lasts forever and is strong. So I'm not a tailor or a seamstress, but I can sew up a bird pretty good.
At the museum support center just outside Washington, D.C., any recently living creatures are cleaned with the help of a non-human workforce. Carnivorous beetles.
They can get in very small nooks and crannies that we otherwise can't reach easily. They're kind of our unsung heroes.
You can't have rotting flesh in storage.
Good job, good job, good job. Thanks, guys, for all of your good work.
Clean specimens are tagged and filed away in uniform lockers. And over the past decade, the museum has also turned millions of items into digital files.
We're about to 3D scan this cactus. Absolutely do hear this in my sleep on a regular basis. If it weren't for the flash, I would be falling asleep at my desk right here, but thankfully it's there to keep me awake.
There are no giant bones, but we have actually 3D scanned other bones as well. We did a skull of the dire wolf.
Sylvia Orley helps lead the digitization effort.
I think, wow, how great is it that I get to handle these specimens every day? I just feel very blessed to have this job.
In 2022, Sylvia's team completed a 7-year effort to digitize the entire U.S. herbarium, a collection of over 5 million plants and flowers. Now they're scaling up to digitize the entire entomology collection, which is 7 times larger. A new scanner, like the setup in this video, will take up this entire room and photograph 200,000 pollinating species a year.
That's going to simplify the process greatly, where we're going to be able to digitize between 1,000 to 2,000 specimens per day using this conveyor belt.
Sylvia says this project will take about a decade. But it might be faster with AI tools, which the team is already using to reposition cameras and transcribe labels.
And I expect in the next few years, there is going to be an explosion of use so that we may be using AI for 50% of our work. But it is just in its infancy right now, so we're excited to see what's going to happen.
But not everything arrives at the Smithsonian intact. And for at least one department, that's entirely the point. In 2023, the U.S. had over 19,000 wildlife strikes on aircraft. almost all of those were birds. Globally, they cost the airlines billions of dollars. The Smithsonian's Feather Identification Lab is here to do something about that. It's run by Carla Dove. Yeah, that's her real name.
I get that all the time. My name is very appropriate.
Every year, her department receives up to 12,000 samples of bird remains scraped off aircraft. It's lovingly called snarge. That comes from US military slang, combining the words snot and garbage. It's up to Carla's team to use the snarge to ID the birds, so airports can use the most effective techniques to avoid them.
I asked my 4-year-old, what do I do at work? And she says, you look at dead animals. Yeah, that's true. That's what I do at work.
Sarah Luttrell is a research scientist in the bird department.
So the start to our day every day is to open these up. We get a little bit of bird. We never know how much. And then we get the data.
Over 70% of bird strikes occur below 500 feet, mostly during landing. Most of these collisions are non-fatal for humans, but if a bird gets sucked into a jet engine, the results could be catastrophic. Remember the plane that crash-landed in the Hudson River?
Less than a minute into the flight, the pilot reported a double bird strike.
In that case, we got 69 bags. Because as they went through and did the investigation, they went all the way into the engine and they wanted to know how far into the engine the feathers went.
Identifying the species of bird helps airports develop wildlife control programs and sensors to avoid collisions. If they can't physically ID a sample right away, the lab can compare its feathers to one of the 500,000 specimens in the museum's collection.
You still need the physical sample and you still need the physical expertise to do these complex identifications.
The team can get a pretty solid ID this way, but they often need extra help.
We can't be 100% sure that this feather came from a Eurasian collared dove and not a pigeon. So we're going to have to send this to the DNA lab.
The Smithsonian's Laboratories of Analytical Biology, or LAB for short, is the nerve center for all things DNA.
Our big goal here is to document life on the planet. And in order to do that, the tool that we're using for biology is now genetics.
Lee White is in charge of this multi-million-dollar genome sequencing operation. A genome is the complete set of genetic material in an organism. Think of it like a blueprint for building and running a living thing. In 2009, Lee's lab helped Carla Dove's team map the genome of every known species of North American bird. That reference database has helped the Feather ID lab work more efficiently. It turns out you only need a few birds to decode the instruction manual of an entire species. Relative to other organisms, birds have a smaller genome.
We only need to do about 5 blue jays from across their range to capture all of the genetic diversity that's in blue jays. So we might have 1,000, but we only need to do 5.
Now Lee's team is working their way through the entire Smithsonian collection.
Every plate you see holds 100 DNA samples from many different projects and many different groups of organisms.
And thanks to new lab equipment, chipping away at this massive task has never been faster. This robot automatically prepares samples for DNA sequencing, a process that used to be done manually.
So this robot's really nice. I can run up to 96 samples at a time. Today we're just doing 8 because that's all I had to work with.
Katie Murphy is a genomics specialist who runs all of the instruments in the lab.
So it'll take about an hour to get through that step, but that's an hour of the robot working that I don't need to be here for normally, although it's really fun to watch it. And one of the big projects going on is doing that for all US fishes. And so right now I'm prepping samples for 8 fishes.
What used to take us a month, we can now do in less than a day. But what we end up with is magnitudes more data.
All that data is helping to build a digital Noah's Ark. It's called the Earth Biogenome Project, a global effort to finish mapping the genome of every species on Earth within the next decade. Once complete, it'll not only help scientists better understand life on a changing planet, but it could also unlock huge benefits for human civilization. This biological big swing stands on the shoulders of the Human Genome Project. A little over 20 years ago, scientists successfully mapped all 3.2 billion pairs of human DNA. In doing so, they sparked a biotech revolution. It gave scientists the tools to map, analyze, and compare genetic information at scale, transforming things like forensics, drug development, and ancestry testing. Biologists expect the Earth Biogenome Project to have similar results. But scientists estimate that 80 to 90% of Earth's species have yet to be discovered. And DNA mapping has revealed that some of the museum's own specimens, once thought to be related, are actually genetically distinct.
As we go into our collections and sequence them, we find undescribed species hidden in our collections. So the goalpost keeps moving. And so we have a long way to go.
Take these little guys.
As of right now, these two fish are considered to be the same species. And they look pretty similar, right? The one in my upper hand is from the Northern Caribbean. The one in my lower hand is from the Southern Caribbean. And because we've sequenced both of them, matter of fact, if I flip them over, you can see the tissue sample that we've taken from both of them. And then we ended up sequencing them. They turned out to be wildly different. And I can't tell you right now exactly why they're different anatomically, but that is exactly what I'm going to do tomorrow, is CT scan these two specimens so that I can learn a bit about their anatomy and be able to tell you why they're two different species.
So even if LAB makes it through the Smithsonian's entire collection, it's just the tip of the iceberg.
Scientifically, we might have done the DNA on hundreds of thousands of species, but there's millions out there. But that's the ultimate goal, is to do it all.
It turns out that analyzing samples they do have helps Smithsonian researchers figure out where to look next. And to do that, you don't need a critter or even a piece of one. All you need is a sample of dirt or water and a kitchen blender. Chris Meyer has studied sea life in some of the world's most remote islands.
No, I don't spend a lot of time behind this desk.
He worked with Lee to develop one of ocean researchers' most important tools, the Autonomous Reef Monitoring Structure, or ARMS.
I'm a global arms dealer. Yes, we sell arms all over the world to our partners. That system was developed to mimic the nooks and crannies you find in coral reefs.
Essentially, it's just a stack of plates, but tweaking its design brought out Lee's inner MacGyver.
Home Depot, hardware store, online, science supply. It's really skunkworks fabrication stuff to help our scientists in the field process the biodiversity. So some of that is inventing things because they don't exist. We're doing things nobody else is doing.
Researchers strategically drop arms to the seafloor. Over time, plants and animals move in. After the crates are pulled back up, scientists meticulously remove, catalog, and photograph everything larger than 2 millimeters. Then they analyze the plates. All that sea life leaves behind a dazzling array of colorful patterns.
When you go back and get it after a year, each plate might look something like that. So they go from being absolutely nothing to fully populated.
We carefully take each plate apart, photograph each plate in a photo mosaic with a 3x5 grid, and we stitch those together to get massively high resolution pictures because I think those will leave a lasting legacy. It's remarkably high resolution, and that helps for the next step, which allows us to use machine learning to train to classify each of these organisms into their functional categories.
By now, some of the algorithms can tell which species lived in the arms just by the patterns they've left behind.
Once all that's done, we scrape all the plates with paint scrapers, and we put it in a blender and grind it all up, homogenize it, make a slurry, a reef's milkshake, and it works amazingly well. I'm pretty much a blender connoisseur, so.
I think this will be the tastiest reef yet. Look at that, it's frothing.
I go to the different stores where you can buy blenders in the world, and the blender is the key tool.
Living things can leave behind slime, feces, or even tiny cells wherever they go. The genetic fragments trapped inside this slushy mix are enough to create a clear snapshot of the ecosystem. It's called environmental DNA, or eDNA, and it's incredibly valuable.
So we get a better sense of which are the winners and losers in these different systems, and what are the best sentinels of those change.
eDNA can also help prevent huge economic losses.
The other big utility of the arms right now is as early detectors for invasive species. Invasive species cost industries and fouling communities literally billions of dollars.
When we visited Andrea Quattrini, a research scientist, she had just used eDNA to discover a new species of soft coral that was less than a centimeter tall.
We basically just looked at the DNA in the water, and that was a sign that we have a new species that wasn't known to be there before.
That's always super exciting for biologists like Andrea.
So if we do collect a new species, I do get to describe it. You really can't describe it after yourself, but you could find probably a colleague and say, hey, I think I collected something new. Why don't you describe it? And you can name it after me. I mean, we don't do that. That's like a, what's the word? Not a, what's the word? Taboo. Taboo. Yeah, it's a taboo.
Instead, Andrea named this new species after her mentor.
Parasphera sclera. I know it's a big name. Anyways.
eDNA collected from arms is already being used to monitor ecosystems in over 3,000 locations all over the world. But biodiversity isn't just something to study. In many cases, it's something scientists are racing to save. Because over the past few decades, there's been a rapid decline in global biodiversity.
It's just inevitable that some of these insect species are going to go extinct, and collections like this are going to be the only place where you can find any evidence of their existence.
Tools like eDNA can help scientists detect species before they disappear, sometimes just in time to learn something from them. And that's critical, because extinction is forever. That's something Rebecca Johnson knows intimately. Today, she oversees research at the museum, but has a special place in her heart for a species on the brink.
I feel the same as everyone does about koalas. I think they're extraordinary creatures. Many people like to work on koalas because they're so iconic. They're biologically very interesting, but they're also really in need of science because there's fairly major conservation concerns for them as a species.
But the Smithsonian collection can be an irreplaceable tool for navigating an uncertain future.
Not everyone has what we have in our collections. In some cases, we might have the only examples of those specimens in the world.
And scientists are already putting them to work, not just for species conservation, but to protect the systems we all rely on.
If you enjoy food, like we all do, you might not know that we have an insect collection that's used every single day to identify potential pests that are coming into the country that may be a risk to agriculture. Similarly, the botany collection is used in the same way. We have a very huge agriculture industry here that this collection directly benefits every single day.
When it comes to defending America's farmland from invasive bugs, Matt Buffington is one of our first lines of defense.
A cicada will be like, making all this noise, and then as soon as the cicada killer grabs them and stings them, you hear the kind of it's a little sad, but that's nature.
In 2019, Matt received an urgent phone call. A homeowner in Washington state had discovered a freakishly large hornet in their backyard, and scientists needed to verify what it was as soon as possible.
And when that specimen was on its way here, I had a number of emails and phone calls asking to contact them as soon as it arrived. I mounted it. That's one part of the process, so I could examine it under the microscope. And then I went to the collection, and that's when it dawned on me. The significance of this is this might be the first record of this species in North America, and this species is detrimental to honeybee. And honeybee is a major part of our pollination system for agriculture. Vespa mandarinia, otherwise known as northern giant hornet, sometimes referred to as the murder hornet.
This invasive species from Asia had the potential to create a $100 million disaster.
Once that comes in and it gets established, you're not getting rid of it.
After Matt's positive ID, entomologists formed a SWAT team, suited up in special sting protection suits, and went on the hunt for murder hornets. Then, a single hornet tagged with a radio transmitter led them to nest 0. America's first established colony of murder hornets.
A few years ago, this was translocated to Smithsonian for an exhibit on invasive species to North America. And it was just in time because of the 202 individuals in the nest, 96 of them were queens. Each one capable of establishing a new colony. So they got them at just the right time.
Nearly 5 years on, and there hasn't been any new sightings of the murder hornet. But these aren't the only things the collections are used for. Fossils can show us how life has adapted to past climate shifts, and unsequenced genomes might hold the key to drought-resistant crops, or life-saving medicines we haven't discovered yet.
If this collection went away, we would lose the knowledge that is the foundation of what we know about biodiversity on the planet.
Preserving that foundation isn't a given. Scientists and institutions like the Smithsonian are facing funding cuts and political pressure. Behind the headlines, that meant real uncertainty for researchers and staff, and long-term risk to one of the world's most important scientific resources. Meanwhile, private biotech companies sometimes promise they can reverse extinction with pipettes and computers. But as any ecologist will tell you, once a species is gone, it's gone. And no amount of synthetic biology can replace the complexity of real ecosystems. The clock is ticking, but if the federal government continues to invest in science and preserve what we have now, we might still shape a different future.
I think the urgency is that the technology is so good now. You can get an excellent quality genome from an old specimen, the kind of specimens that we do tend to have in this collection, and the questions are really urgent. And so not only do we have the technology to generate the data, we have the need to answer those questions with that data.
So what about the internet rumors about those giant humanoid skeletons?
We have a very large file of people asking that question.
But when we cornered the museum's top director, he set the record straight.
And where are the giant bones?
They're sloth footprints. People thought they were human footprints. And that started the whole Sasquatch giant thing. But we can't get rid of it.
Miners discovered this 8,000-pound crystal in Arkansas. It was donated to the Smithsonian National Museum of Natural History, which has one of the largest mineral collections in the world.
We knew we had something very special.
But donations like this don't happen often anymore. That's because crystals are getting expensive. Really expensive.
Even millions of dollars for a single specimen.
Things that were 50 to 100,000 back in the 90s could easily be $500,000 to $1.5 million today.
But because crystals formed millions of years ago, there's a limited amount of them. What we dig up is what we got. Take the scarcity, growing demand, and that Instagram aesthetic.
They're extraordinary because the uncut ones are just beautiful in their rawness.
And now you have private collectors scooping up crystals at skyrocketing prices.
Our business has been going up at a fever pitch pace over the last 2 decades.
It's all making it harder for museums to get a hold of and preserve specimens like this. So how did the industry get so big? And how did the Smithsonian land such a rare piece? To find out, we followed the journey of this giant Arkansas quartz.
Whenever you find a big one, you only find just a little bit at a time.
That's Josh, a 5th-generation miner. He and his grandpa found the Arkansas quartz in the summer of 2016. Most crystals have 2 sides, forming a pocket in the middle.
And the back of it's just flat. So when you're following that ledge, they're growing toward each other. Their points are basically intertwined. So when we're digging them, we didn't know what it was until we brought it up and sprayed it off.
First, they removed only one side of the crystal.
When we pulled it out of the ground, there was so much mud on it that it didn't even look like a crystal. My grandpa was disappointed. He said, it's just blank. And what we mean by blank is it's just quartz, there are no points on it. And that was kind of discouraging, so we put it on the truck and drove it up here to the cleaning shed and started spraying it off. And we're just like, oh my God, like, this is phenomenal. You know, it was way more than what we expected it to be. And we actually left the other rock still sitting in the ground and went back about an hour later and got that piece.
Altogether, it took them about 2 months to unearth the giant crystal.
That's the biggest one I've found, yeah.
The Arkansas quartz weighs as much as this forklift. The Colemans knew they had to eventually sell a crystal of this size, and the best place to do that is Tucson.
Tucson is the world's largest gem show.
Every year, 4,000 vendors descend upon the city. It's estimated that altogether, $5 billion worth of crystals are sold at the show in just 3 weeks.
So we actually took both pieces to Tucson. The smaller piece, which is the top piece, never come out of the box. The bigger piece, which is the bottom one, we actually had to dismantle our whole store place in Tucson just to be able to get it in there.
That's where the donors found it.
I'm always looking for stuff for the Smithsonian, but probably most of what I find, they go,
We already have that.
we already have that. I knew right away from the size of crystal, it looked like about 8 feet by 10 feet.
You can get a bunch of mineral crystals and glue them together, but to pull up a solid piece like a wall like this, that is rare.
So real estate developers Michael and Patricia called up the Smithsonian.
They said we would love it, but it's not something that we can afford.
Because it's a government-funded museum, the Smithsonian can't use federal money to acquire specimens. So the world's largest national history museum relies heavily on donors like the Burns to expand its collection.
At that time, it was something that we couldn't afford either. I started negotiations with the miner. It took me a few months.
The Burns wouldn't tell us how much they spent on the crystal, but Josh told us they had crystals this size appraised for $4 million and $7 million in the past.
What we got out of it was basically that we're keeping an American treasure for the American public.
But moving it 1,000 miles wasn't an easy task.
They're fragile. With that particular piece, half of it was big pointed and the other half was little bitty points. And those little bitty points, they'll come off real easy.
They suspended the pieces with straps and forklifts and then moved them into 2 wooden boxes.
We have big 55-gallon drum trash bags full of just wadded up newspaper, and it does a great job.
Josh and his family drove those boxes halfway across the country to D.C.
Although we bring a lot of specimens into our collection every year, almost never do they come in a big truck like this. So this is a special occasion for us.
That's Jeff. He runs the gem and mineral collection at the museum.
He makes sure the pieces are in good shape before the museum takes ownership.
Yeah, we're just looking for anything that's kind of gotten loosened up. I mean, everything looks like it's been packed well. Look at this. Wow, isn't that nice? We have at least a couple different generations of growth. It's kind of fun to explore it because you see different things. I don't see anything that's been damaged.
And this is Charles. He's in charge of moving the crystals, and he's been doing it here since 1995.
So this crate contains the first of the 2 Arkansas quartz. This one is the larger of the 2, approximate weight of 5,000 pounds. And there's a smaller crate that has the second specimen, and it weighs 3,000 pounds. We're gonna unload and install in the museum today. Just ready to get it moved, get the job done.
It takes a team of 5 riggers, steel plates, and lots of straps to safely move the crystal.
How much work does it take to coordinate all these people and all?
I don't want to talk about that. Safety is an important factor when you're moving stuff like this. I've been doing this a while, so I am perspiring a little bit, but I'm good.
The display is only 400 feet away from the loading dock, but it took 7 hours.
Finally, by midnight, the quartz crystal is secured in place.
We're thrilled that it's in there. Not everybody gets in the Smithsonian, so for it to go in there is kind of like a kind of confirmation to me that, we're the best.
But in the gem and mineral department, getting donations like this is becoming rarer because of the growing and lucrative business of crystals.
The fascination with crystals has been there since antiquity.
Ancient Egyptians buried their dead with quartz and used crystal in jewelry and even eyeshadow. In China, jade was believed to grant immortality and was built into burial sites for the royal families during the Han Dynasty.
And then the Greeks take it up and had medicinal purposes or could ward off the evil eye or things like that.
It's that spiritual connection to crystals that made them perfect for the self-care world.
And it sort of transitioned in the mid-90s with both the metaphysical boom that started in the 80s and a new group of collectors who were coming on the scene, and they were strictly looking at them from their beauty factor.
It checks so many boxes in that way, right? It can be a spiritual practice. It can also be for wellness. And then you can capture that in media, right? In Instagram and Goop.
Katy Perry, Kylie Jenner, and Gwyneth Paltrow have praised crystals for years. Adele swears by hers for helping with performance anxiety. Today, the industry's worth over $1 billion. Mines like the Colemans have become a tourist attraction.
So you can come out, dig for your own crystals. We've got a zip line that goes over the crystal mine, and we do tours of the mine, and over the past 2 years, those have just took off.
And big beauty brands have gotten into the game.
Interior design, people buy them and set them around their houses.
In New York City, crystal healings for chakra realignment sell for $250 for a single hour-and-a-half session.
Crystal healing operates on the vibrations of your body and, taking any out of balance vibrations and raising them to be at their natural state.
Different crystals supposedly offer different types of healing.
But there's no scientific evidence to support crystals' healing power. A 1997 study from a Goldsmiths University of London professor found that crystals' power came from nothing more than placebo.
Still, that hasn't stopped consumers from scooping them up.
It's just now it can be commodified, put in social media, Instagram.
All this popularity has made the competition for crystals incredibly steep.
Science people are still into it. You have the metaphysical interest that's driving it, and you have the art crowd. Through that growth, you have appreciation and value.
Daniel said he's seen crystals sell for 100 times what they did 20 or 30 years ago.
Many of the mineral specimens that you see in these cases and on display sell on the market these days for tens of thousands, hundreds of thousands, even millions of dollars for a single specimen. And so the art world and the mineral world have actually sort of become a very similar kind of an economy.
It's difficult for publicly funded museums to get access to great material. And that's because the private collectors today are so fiercely competitive and have such access to money that they're buying all, or not all, but a majority of the great objects that are out there.
I have no problem with people collecting these things, but the difference is once they're here, they're forever in the public domain, which means that we can put them on an exhibit for people to see them. Plus, it provides access to scientists, so it's not just a private collector looking at it in his room or basement or her basement or whatever.
And what makes this Arkansas quartz so worth preserving is its rarity, both in size and existence, because there are only a limited number of crystals in the earth.
The crystals formed probably 300 million years ago.
Deep in the Earth's crust, and they need the perfect conditions.
Crystals can form either from magma cooling down slowly, in watery silica-rich environments, or under intense temperatures and pressure. But all crystals need an open space to grow.
And then as they move up, through erosion and through outcropping on the Earth's surface, we're able to then find them. Somewhere deep in the Earth right now, probably 10 kilometers down, that we'll never see in our lifetimes or probably in another 50 lifetimes, there are minerals forming, but they'll never be available to us.
But none of that seems to be slowing down the industry.
I think the demand is just going to continue to, increase.
You are the first person to see a bit of Sue's blood vessels.
Yes.
That's
and
rad,
you're
isn't it?
gonna be the second. It's this hollow branching shape.
That Sue in question is Sue the T-Rex, and we're about to see proof that she was warm-blooded.
We went to the Field Museum to see how they work their magic. Our first stop was...
So here is our dinosaur and oversized collections.
That's Jingmei O'Connor. And she, you know what? I'll let her tell you.
Yeah, I would say I'm one of the world's experts on Mesozoic birds. Not to brag or anything, no, I'm just kidding.
And one of the key questions she asks with her research is why birds were the only dinosaurs to survive the Cretaceous-Paleogene extinction.
We need to look at birds, and we need to back up from birds and look at the dinosaurs closely related. I think it's kind of funny, but we have a drawer full of bits of Sue. When you have the edge of the puzzle, that's the easiest part to do, right? The inside of the puzzle is harder to put together. So these are like the inside puzzle bits. You can't figure out where they go, but this is to our advantage because these are the type of fragments that it's okay for us to do destructive analyses on.
The destruction of these rare and one-of-a-kind fossils can involve slicing or dissolving in acid. Slicing allows the scientists to study specimens histologically.
You have to cut a piece of the bone, remove it, grind it down so it's really thin so that light can pass through it so you can study it under a microscope. It's a cost-benefit analysis, essentially, right? What are the questions you're trying to answer? Is it worth it to damage this extremely rare, important fossil?
To show me how it's done, they demonstrated with a prehistoric bird bone.
You're going to bring it to, in our case, Akiko Shinya. She's our chief fossil preparator. She's amazing.
Akiko starts by taking a small slice of the specimen.
You then take your little chunk of bone that you removed and you drop it into resin, and then this needs to cure for several days. I mean, can you imagine playing like Dungeons and Dragons with like dinosaur dice? Yeah. Exactly.
Then you glue your D6 to a microscope slide and slice it down even more.
I did a terrible job with the saw. I almost destroyed everything with the saw, actually. I feel like I'm pushing too hard. Okay. I did it. Still have quite a bit of ways to go, so yeah, it will take a while.
At this point, it's too small to slice, so Akiko will grind away extra layers.
Rim here is the diamond dust, so that's where the grinding surface is. If you bring it tiny bit, a little by little, and then you will eventually start hearing the noise.
We're listening for that shoog shoog sound. The slide ultimately needs to be between 30 and 80 microns thick, thinner than a sheet of paper. Akiko basically uses finer and finer grinders, like varying grades of sandpaper, to slowly shave off extra layers.
Straight down, that's the key.
I will try my best.
Yeah, I'm sure you can.
You scared me now, though.
There's a polishing stage to smooth out any major imperfections that might obstruct the sample.
You're not getting away from me.
And a second polish for fine-tuning.
This one feels like someone is like pulling carpet out from underneath the block.
Even with your naked eye, you can learn about a specimen. Here's a piece of Sue's rib, and you can see how Sue grew, almost like rings on a tree.
You see these faint lines?
Yeah.
Yeah, it'll be
It
much
looks like agate.
Yeah, it'll be much clearer once we get it under a microscope, but those are the lines of arrested growth.
Do you want to look in?
Yeah, okay.
And here we're looking into the rib of Sue the T-Rex. You notice that the space between these lines of growth is becoming smaller and smaller. So when it was younger and really having to bulk up, right? It was growing very quickly, and as it reaches adult size, growth slows down.
The thinness of these sections was surprising because it shows a fast growth rate, a key indicator that Sue had a high metabolic rate, meaning she was probably not as cold-blooded as scientists previously thought. Scientists also looked at another indicator of high metabolism, which is actually color. More diverse colors in a species tends to mean a higher metabolism.
Here we are looking at an SEM image of a sample from a feather of a 130 million year old bird called Ao confuciusornis.
Pre-extinction.
So the only fossil bird older than this fossil bird is Archaeopteryx.
A scanning electron microscope relies on electrons instead of light to magnify even more detail.
And so if you look closely, you'll see these, this is literally what we call them, sausage-looking structures. They are aomelanosomes. So aomelanosomes are responsible for the color black.
Melanosomes are organelles found in animal cells that are associated with different colors. When they fossilize, they leave behind distinct shapes.
If they're very nicely aligned with each other, we can tell it's iridescent black. If they're kind of more oval-shaped aomelanosomes, that's gray. And then if it's a phaeomelanosome, we call these ones meatballs. Literally, this is like in papers, they're like the meatball-shaped ones. I'm hungry. The meatball-shaped phaeomelanosomes are responsible for a rusty red color.
Many of the genes responsible for melanosomes are also linked to things that affect metabolism. So evolving one most likely evolves the other. And with both meatballs and sausages, Eoconfucius ornis shows way more melanosome size diversity than modern-day cold-blooded lizards.
So we can say that the dinosaurs that are becoming smaller, that are getting these large, extravagant ornamental structures that are then able to evolve flight are also becoming more colorful.
But these melanosomes can only tell us so much. They're an indicator of warm-bloodedness, but not definitive proof. This is where Yasmina comes in.
Everything that Yasmina is doing, 5 years ago, that didn't exist.
I'm a molecular paleobiologist. My passion lies within the clade of dinosaurs, including modern birds. People tend to think of bones and shells and these kind of like hard tissues that preserve much more readily. But if we want to get a complete picture about the diversity of life on our planet, we really depend on soft tissue preservation.
Soft tissue is the squishy stuff like skin, blood vessels, and other non-bony materials that scientists didn't even think could preserve until recently.
So about 30 years ago, vertebrate paleontologists tried for the first time to extract soft tissues from dinosaur hard tissues.
Her name is Mary Schweitzer, and people did not believe her findings.
It was very critically perceived. And people thought for a long time that while these soft tissue structures very much look like the original biological structures, they could not possibly be related.
But soft tissues do preserve.
Why do they preserve? This is absolutely paradoxical based on what was known scientifically at that point in time.
You can actually see traces of it under a UV light.
What we're gonna do is just shine the light and look for things. It looks like some of these may be scales that are preserving soft tissue, not all of them.
Once soft tissues are suspected, demineralization will isolate them if they're present so you can see the structures. This is where I got to try something with Sue's bones that has never been done before.
So you mentioned that you would like to dissolve a Sue fragment and help us look for organics, because every bone is going to have the tissues that we're looking for if they are in fact preserved. But Sue has exceptional preservation, so we are quite hopeful.
We're taking this bit of Sue and dissolving it in hydrochloric acid.
So for example, like if you drop a little drop on your skin, it's gonna draw the calcium out of your bones.
Just leach it straight out? Yes. And leave a divot? Or...
Oh yeah, it's gonna be like a huge edge mark. Yes.
That's
which is
wild.
Why hydrochloric acid requires a lot more ppe.
The acid will dissolve any inorganic rock, but leave behind the organic soft tissues.
Starting to look like a hazy IPA. You seen anything?
It's all still in suspension. Yeah, we'll have to.
Me too.
Give it a little bit of time.
After about 15 minutes, the precipitate settles and Yasmina pipettes it onto a slide.
From here we go to the microscope.
Let's go.
Ooh, and this is looking good.
What are you seeing?
We have a couple of extracellular matrix pieces, blood vessels, large blood vessel fragments.
You are the first person to see a bit of Sue's blood vessels.
Yes.
I was the second person to see Sue the T-Rex's veins.
Right now, take a look.
Okay.
You see the blood vessel structure is right in the focus center. It's this hollow branching shape. That is definitely one of the bone vascular canals.
Oh, I see it real good now. Hold on. They've just been in there the whole time.
They've been sitting there
around.
for 65 million years.
That was rad.
Because we can look at this, are of course fascinated by the fact that soft tissues preserve in deep time. But there's actually a lot of information in the molecular composition of these materials.
Demineralization shows us that there are soft tissues present in a specimen, including the proteins, lipids, and sugars that indicate a high metabolism.
But to concretely say what soft tissues are present and prove that this vein is really a vein and that these metabolic stress markers are actually here, we need to study the chemicals found in the tissues. This requires a brand new method, an application of Raman spectroscopy developed by Yasmina and her team.
It's the brightest, purest green that you will ever see.
Yeah, why the green light?
We're using a green light that is exactly 532 nanometers that is particularly good for characterizing organics with many unsaturated carbon bonds.
It's a non-destructive technique, so you can stick entire specimens under the laser without having to prepare them at all. We popped in a piece of Sue taken from her femur core.
We want to make sure that we're looking at a spot where we don't have too much surface texture, where we have a lot of carbonaceous material preserved.
The laser excites the different materials in the sample.
And so these chemical bonds, they start to vibrate in direct response to their chemical environment. And these very specific little wiggles are then detected in form of a spectrum. Now, if we want to, for example, learn something about the metabolic rate of Sue, we collect, the spectrum for Sue and for all other kinds of dinosaurs that have this mode of preservation. I can show you basically what it looks like when you compare these different spectra.
This is Sue's Raman data compared to data Yasmina got from other dinos.
So every individual line basically represents evidence of the total composition of one of the fossils we've looked at.
So these numbers are sort of different wiggles?
Yes. So you get a different wiggle at each of these different wave numbers, and that then evidences a different kind of molecular bond vibration, so different kind of chemical compound present in the sample.
Different compounds tell us different things about the dinosaur we're looking at. For example, thioethers, sulfur heterocycles, and nitrogen heterocycles in the soft tissues signal high metabolic stress.
We have very high amounts of these metabolic stress markers in warm-blooded animals, and we have very low amounts of these metabolic stress markers in cold-blooded animals. This peak here, in combination with this peak here and this one here, tells us if we're looking at a warm-blooded or a cold-blooded extinct animal.
This wiggle here is proof that Sue the T-Rex was truly warm-blooded. And it once again changes the story of evolution that scientists have been telling and retelling since the early days of paleontology.
It has been suggested that the high metabolism of birds eventually helped them to better adapt to the changing environments, the changing conditions right after the mass extinction event. What we basically realize here, once we start putting these new physiological data in a context, is that this incredibly high avian metabolism is actually not an avian innovation.
It's not just Sue either. Yasmina's work has definitively found that most dinos were warm-blooded, practically upending what was once common knowledge.
I have been coming to the Field Museum since I was a little kid.
Oh, the Tully monster! This is my favorite fossil. I won't get distracted.
Being able to come to the Field Museum and see how the Field Museum works was like a dream come true for tiny paleontologist Abby.
Here we go.
This is lava. And this is also lava. One's man-made and one's, well, coughed up by Mother Earth. But both these scientists are working toward the exact same goal, figuring out how to predict the unpredictable.
To be able to make the best decisions about how to keep people safe, it's important to be able to predict what is lava going to do once it starts flowing out of the vent.
1/10 of the world's population lives within the danger zone of a lava flow, which means knowing how lava works and reacts to elements like water, metal, and ice is key. So, on this episode of Science Skills, we're going to look at 2 ways scientists study lava, starting with DIY.
This huge furnace behind me is Syracuse University's personal volcano and brainchild of an unlikely scientist and artist duo.
That's Professors Jeff Carson and Bob Vysotsky, and they didn't feel like waiting for a volcano to spit up lava. So they decided to make their own.
The project originated really when Bob came into my office and said he wanted to make lava. I thought that was pretty crazy, a suggestion at that time. But the more we talked, the more we saw that he had a really good idea of what needed to be done.
And the first thing that needed to be done? figure out what to make the lava in, which is where this came in. The tilt furnace is really the statement piece of the whole operation. It can hold hundreds of kilos of lava and execute experiments about viscosity, morphology, structures, and formations. But she's a little bit finicky when it comes to lava making.
The furnace literally melts itself and tears itself apart over a very short period of time.
These were originally made to melt bronze and aluminum, but the Lava Project has repurposed one of them to melt up to 800 pounds of billion-year-old basaltic rock shipped all the way from Wisconsin. The process takes hours. Bob and his team pile the rocks into a receptacle called the crucible, turn up the furnace, and gradually bring the rocks up to temperature.
If we were doing just lava and melting stuff, the furnace would be on about medium, and we would just never turn it down or up.
What temperature?
It just runs. Medium is a sound out there that I hear the flame. I can adjust the furnace blindfolded and tell you what it's doing, and it's all sound. There's just a butterfly valve in here, and
oh, you can hear it.
That's it.
We looked this up later. Medium is also somewhere between 2,000 and 2,400 degrees Fahrenheit. So pretty darn hot, which means these scientists really have to suit up. These suits made of aluminum can withstand radiant heat up to 3,000 degrees Fahrenheit.
We used to wear welding leathers, but it dries out from the heat. When you start to smell barbecue, because it's pig skin, you knew that you were too close to something because you're cooking, your clothing is cooking.
How do you know you're too close with these guys?
You don't.
You don't?
These are the spats, the apron, which I wear around my waist, the jacket, which, put your arms out, right? Because you don't need it in the back.
No. It's like a campfire style.
Exactly.
Back
and it's
heavy
just
is
that.
cool, that's warm.
So there's that. And then the helmet, it looks like it's a regular tinted thing like sunglasses. But this is 24 karat gold. That is a sheet of it.
What is it about the gold? Is it just the reflective quality?
It's so highly reflective, and it's why you see satellites and stuff, why they have the gold foil on it.
And this is really similar to some of the stuff that like volcanologists would use in the field, right?
Same stuff.
Yeah, but maybe with a back?
They have a back on it.
Yeah, in case the volcano's behind them. Do you want to show us how it works?
Yeah, let's go talk about this stuff.
Do we need any of the gear?
It is sweltering. How hot is it up here?
Well, the bright yellow you see back there, that's about 2,800 degrees Fahrenheit. So right now the lava in there is too hot. When we dump it out of here, it's about the meter it falls from the spout to the trough, and through the trough, we lose about 275 degrees Fahrenheit. By the time it hits the end, we want to be at 2150 Fahrenheit. And that's the magic spot for the lava.
Researchers are looking for that sweet spot between 1,600 and 2,200 degrees, the range for natural lava. Knowing the lava's temperature at what time and where is crucial. So the team has an array of 10 digital cameras to capture 3D images of the flow, and a thermal camera which can read up to 3,600 degrees Fahrenheit. That way, researchers like Ariana Soldati can analyze both the lava's movement and temperature, leading to a key piece of data.
Viscosity is possibly the most important property in volcanology. It really controls everything from eruptive style to appearance of the flow. And the main physical property that controls viscosity is temperature. The hotter something is, the less viscous it is, and the cooler it is, the more viscous. So it's really important that we can tell what temperature the lava is, because we want to match that with the viscosity.
With this, the team can study how different variables like metal or crystals affect how fast the lava cools and therefore its viscosity. But there's the lab, and then there's the real world, where unplanned and unpredictable factors come into play. That's where this guy comes in.
Well, this is a piece of the Earth that we call the mantle.
That's a piece of the mantle?
This is the piece of the mantle. And this is one of the sort of sidelights that makes some volcanoes incredibly important to study.
This is Ben Edwards, and he likes to get lava data straight from the source. Here's him collecting a sample from a flow in Russia back in 2013. As you can see, Ben's protective gear has more coverage than what they use at Syracuse. because sampling from a natural lava flow can be a 360-degree experience.
If you're going next to a lava river to sample, even in this suit, like I was doing this in Russia from a lava river that's maybe 10, 15 meters wide, and after being there for a minute or so, making some measurements, I could hear my Russian colleagues saying, "Ben, move back, you're smoking." But it was getting hot enough in the suit that even after about 30 to 45 seconds, I had to back up.
When Ben's around to witness an eruption, he's prepared to collect data, a lot of data.
Am I going to focus on taking lots of lava temperatures? Am I going to focus on getting lots of samples of lava? Am I going to focus on using drones and trying to map very carefully how fast the lava is coming out?
To pull a sample out of the flow, Ben usually uses a rock hammer. But
if I was trying to collect really hot samples, I would probably use some sort of an iron bar that wouldn't catch fire. Like this is okay for short. It's made of wood. Yeah.
Here's a clip of Ben's colleague, Alexander Belousov, using an iron bar to collect a sample.
He rests the bar on top of a rock and he uses a lever to pry the sample out because it's kind of non-intuitive. It's a lot stickier than it looks if you're just watching it flow by. It's like, wow, that must be pretty fluid because it's moving pretty fast.
That dollop of forbidden honey is then dunked into a bucket of water, not just to cool it down, but to cool it down fast because
as the sample cools, naturally it does produce these crystals.
The crystals, yeah.
And if you want to see what was in the sample as it was moving down the lava stream, then you want to cool it like that to kind of take all the heat out and basically turn the heat off so that you kind of preserve the sample. And you preserve the crystal content and the sizes of crystals that were actually in the active lava flow.
Crystal size impacts viscosity, so extra growth would lead to inaccurate measurements. To take the temperature of that flow, Ben might use a handheld FLIR camera, like Ariana did in the lab, or a 4-channel data logger.
Each one of these yellow things is a separate thermal probe. So with this recorder, I can record 4 temperatures at once. For example, if I'm interested in figuring out how fast the lava is cooling, right? So here's my lava surface. I might want to put one of these in, just barely in, and the other one I might want to have a little bit deeper. So I can put 2 of these together, and I'm measuring different temperatures now in that same cooling surface.
But you probably won't get those probes back.
I've got wires that are buried in Kamchatka because, once you get 2 feet of this underneath the lava flow, you're not going to get it back out.
That's not yours anymore.
No. It's one of the great things about the Syracuse Lava Lab, right? Because I do a lava flow there, and in the end I take my big hammer and I recover my equipment.
If you don't have probes to spare, you might try thermal mapping the flow from above.
And so drones are really revolutionizing what we can do to study active Earth processes.
You can strap a FLIR camera, a regular camera, or gas sensors to a drone. Potentially even all 3 if you get a drone big enough.
That basically become a volcano observation platform as opposed to just a drone. And one can envision even someday a drone that would have some sort of a tool that would hang down that would allow you to, if not sample lava, because it is tough to get your little sample bucket out. You wouldn't want your drone to get pulled into the lava flow. But you might be able to catch volcanic ash. You could hang a big piece of duct tape that's 20 feet long from the drone and fly it through a dilute ash cloud, and some of the ash particles would stick to the duct tape.
It's just like a fly trap.
Exactly.
But until robots officially take over, we'll need humans on the ground risking their lives and arm hairs to study lava flows.
It's like the lava domes of Montserrat. The only reason we know there have been 3 or 4 domes, I can't remember which, is because there have been people watching and sampling. And yep, there's a dome, and then boom, the dome blew away. Oh, there's another dome. Then it blew, right? And if there wasn't someone there to watch, we might not necessarily know.
So it's important to be in the field for posterity?
Well, and for science, right? If we're trying to understand that volcano and what it does over time to predict it in the future, and that's the challenge we face when we go to older volcanoes and try to understand what we see in the older volcanoes, because there was no one there watching.
Data gathered in the field helps shape safety plans for people in specific regions. But applying those learnings around the world would be almost impossible without careful testing in the lab.
As geologists, we always need that starting point of what happens in the field, what happens in reality. But unfortunately, in nature, there's no repeatability. Every time there's a lava flow, every time there's an eruption, it's going to be different. You have no control over any of the parameters. Here we can vary things in a systematic way, and this allows us to isolate what could be the cause and what could be the effect and tie them together.
Roasting marshmallows is an art form.
I would say it's a science. All right.
This gray 3D printed face is my actual face, but this white one is based solely on my DNA. That's not what I expected at all.
It's not an exact match, to say the least, but forensic scientists are tirelessly improving their ability to identify people without ever seeing their faces. Why? In criminal cases involving unidentified remains, recognition can be the last chance to develop leads after traditional methods fall short. And if they're lucky, giving a victim their identity back could close the case. That's what happened in 2017 when a company called Parabon created this facial reconstruction. The image got 26-year-old Shaquana Caldwell recognized, and in turn caught her killer after months with no leads. Parabon combined two methods of facial reconstruction: forensic sculpture and DNA phenotyping. Forensic sculpture uses skull remains and population samples to create an idea of what the victim looked like. But this method won't ever be 100% accurate. DNA, on the other hand, could get us as close as we can to a copy machine. That's how we ended up at Shriver Lab at Penn State.
This is just envelopes and envelopes of hair.
In any other place, this cabinet full of hair would be really weird.
They're collecting data about people's hair type, facial features, and more to build an algorithm to take my DNA and spit out a replica of my face.
Does everyone sound like this when they do it?
I'm nervous I'm not going to recognize myself when I see it on the screen. Not because it's not a good reproduction, but because I have a different vision of myself than reality.
Ready?
I'm ready. Okay. That's not what I expected at all.
This final image is a composite of my genetic info and the collected data from people who share my genetic ancestry. Right now, the algorithm works best, meaning the predicted face is most recognizable when a person looks, well, statistically average.
So you can get ancestry axes estimated from the genotypes, and then we layer on top the feature-specific genotypes.
Gotcha.
There are some big differences between my actual and predicted faces.
Which is telling me that we need more information in the model.
Humans share about 99.9% of DNA. But what makes me look like me is in the last 0.1%. And that's thanks to single nucleotide polymorphisms, the variations in base pairs throughout our genes.
This is easiest to see with our eyes. I have 2 adenine at the base pair most associated with having brown or hazel eyes, which I do. Now, if you take the A's out and you replace them with 2 guanine, then that likelihood shifts towards blue or green.
Mark and his team are building a database of which SNPs affect which features.
We're really at an early stage. I mean, we have 200 genes that have significant effects on the face.
At the moment, their formula isn't perfect. A lot of traits are controlled by multiple SNPs, so it isn't a simple one-to-one connection.
You know, the distinctive features that we really rely on to identify each other are not necessarily coming through.
Small details are missing, like my cheek mole, and my nose is a bit pointier in reality.
I don't usually think I look like my dad, but without the mapping, the texture mapping, I think this looks a lot like him.
And those are probably genes with big effects, because you know those differences that you see that make somebody distinctive often stand out in their siblings. Like you said, your face looks like your dad.
Plus, SNPs are associated with different traits, not definitive proof of them.
Like, I have all the right SNPs for not having a cleft chin, but tell that to all of my bullies who called me butt chin growing up.
With so much variation, to get this closer to this Mark and his team need data from as many people as possible.
And so if you already have our DNA, what are we getting here?
In order to inform our research, we need to take measurements and data on many phenotypes. The idea is that hopefully by collecting all of this data and associating it with what you really look like, we can then use that to inform an algorithm to predict on someone that we don't know this information about.
So I decided to offer up my data to the algorithm.
All right, snip away.
Hair can make or break a case, but the most common form of forensic hair analysis isn't scientific.
The most common forensic form of hair analysis often uses racial categories to categorize hair. And so in this way, we're able to study hair more objectively. So we're going to embed your hair in a low melt point plastic and then cut that embedded chip in half, and that'll allow us to look down the shaft of the hair. And so we call that cross-sectional. And we can look at the elliptical nature of your hair, whether it's more circular, the thickness, see if you have a medulla, because some people don't have a medulla in their hair. The medulla is the center portion of the hair. You can kind of think of it as like the bone marrow of the hair. This is really important forensically because if you are a forensic scientist, you get up on the stand, you say it's an African hair type, then you're going to sway the jury into thinking that the perpetrator was a specific race.
They also sample your skin pigmentation.
So what this does is it measures the melanin in your skin. I'm gonna need 3 parts. I'm gonna need this part of your arm right here with your palm facing up because it's not exposed to a lot of light. We're gonna need your forehead because that's exposed to a lot of light, obviously, and we're gonna need your hair because we're gonna be also looking at your hair pigmentation.
This little thing is sort of like what they use in makeup stores.
Yep, that's right.
For weight and BMI, the Danita scale shoots a small amount of electricity through your body.
Okay, we're gonna electrocute you.
Okay.
Small.
What about my mic pack?
It should be fine.
Okay.
You don't feel it.
I know, I'm just nervous.
Because I'm a participant in the research, they need to know what my actual face looks like.
So this is essentially our low-tech zero-gravity machine.
Well, I feel it. I'm in space.
Gravity affects how your face lies on top of your skull, so getting photos from all angles provides a more accurate final product.
Basically what we'll do is we'll take all the photos from all the angles that we took, put them into our vector software, and actually stitch them together. So we'll get something that you can actually revolve around on a computer screen.
You're going to put this up next to the recreation from my DNA and see how they match? Or
yeah,
don't?
we're going to see how accurate we can predict your face.
By using known faces to check their work, they can hone the accuracy of the algorithm when predicting unknown faces. And the more times they see SNPs correlating with different traits, the more definitively they can connect them.
Do you have to sample everyone on Earth to get this perfectly accurate?
We don't need to sample everybody to understand how something works. We need a representative sample, ethnically or population appropriate. I wouldn't take our methods and the data we've collected and go to Finland and say, okay, let's set up a big phenotyping lab here without sampling Finnish people.
There's still a long way to go before this technology can become mainstream.
One of my deep concerns over the forensic applications of molecular photo fitting are the premature deployment of the methodology. It's important that investigators understand what you found and will use that information that you found appropriately. Otherwise, you give the investigator an image of what the person might look like, and they can say, well, that image is not the same as my defendant.
The team needs to collect more data, improve the computational power of the algorithm, and simply more time to get things right. But even then, DNA is just your blueprint, not the final product. As we mentioned, SNPs rely on probability. Plus, there's plenty of environmental factors that come into play.
We don't expect every feature is predictable, even identical twins.
It can't tell us things like a person's gender, if they have scars, or if they dye their hair or wear makeup.
Some might enjoy the sun much more than the other twin, and you can see those kinds of differences emerging because of the environment.
The key to using DNA phenotyping to solve crimes isn't perfecting the tech. It's understanding its limitations.
This has been a 20-year project already, and I expect we'll continue in the next 5-10 years. We'll have more than double our current sample size and be able to say a lot more about appearance.
Thank you so much.
Sure.